mitosis. Mitosis is where we get the typical cell divisions that are producing new cells in our bodies. And we'll look at part of what happens there, and then I'm going to let you out early. So we'll probably do this till about 10:30 and then call it good and consider ourselves a little ahead of schedule. Now, the test that you have next week is not going to have anything on cell division. Even though we will have talked about it some today, I'm not going to have it on next week's test. Next week's test will be, as I said, looking at materials that I had on the syllabus as assigned in weeks 3 and 4, where we've had part of chapter 4 on the membrane transport, all of chapter 5, a little bit of chapter 6, and all of chapter 7. Those will be the materials that are on the test we're doing next week. I will post that on Canvas so you have it more as a hard and fast document to look at when you're doing your studying for the next test. So, questions before I start in here for a little while on cell division. All right, let us look some at cell division, specifically looking at mitosis. So mitosis is the typical kind of cell division that happens when we have growing tissue, or maybe if you're repairing skin damage. So what you're doing with mitosis is you're making copies of cells. You're starting with what we call a parent cell and producing 2 daughter cells. Doesn't have anything to do with whether you yourself are male or female. The cells that are being produced from a division we call daughter cells. And what happens with this division is we are making exact copies of all the genetic directions. We're duplicating the nucleus. We're duplicating the DNA that's inside the nucleus, and then splitting to make 2 cells, and each cell gets 1 complete copy of all the genetic information. So if you have a bunch of chromosomes in the parent cell, a bunch of DNA molecules, and they all give the genetic code, you make copies of all of those chromosomes, and then mitosis is a very precise way of getting 1 copy of each of those chromosomes put into each of the new cells. So every new cell knows how to do all the same stuff that the parent cell did. The genes are telling the cells how to do things, like how to make their proteins, and the daughter cells inherit an exact copy of that same information. It's an exact copy except when a mutation happens. We'll look at mutations later because they do happen, but they're not very frequent. And we'll start off initially treating this as if it works perfectly every time, because most of the time it does. So the DNA gets split up and divided exactly after it's all been copied, along with the cytoplasm. Now, the thing with the cytoplasm is the division there might be unequal. The nuclear division is equal, but the rest of the cytoplasm may actually divide to produce cells that have different sizes and shapes and actually go on to do different things. So just because the cells have the copies of all the same DNA, it doesn't mean they have the same future. As we are developing from an embryo, we've got cells that are specializing, and these parent cells are producing daughter cells. Different daughter cells may have different jobs, but they all have copies of the same DNA. So what's happening with mitosis? In us, it's serving for us to grow. We all start out from a zygote. That's the original fertilized egg, where one sperm cell fertilizes 1 egg cell, and that zygote has a copy of all the genes that will then be copied throughout our bodies in all of our cells. So we grow up from that zygote with division after division after division, going from 1 cell to eventually around 50 trillion in the adult human. You're growing, you're having the organs develop, the different tissues develop, you're repairing damage along the way. Mitosis is happening basically in every tissue that is growing as you're approaching maturity. And it'll continue to happen to maintain tissues in some areas. Other areas it will stop. So for instance, when your brain cells have reached maturity, they stop dividing and you're not making more neurons. The neurons are the big communicating cells that are in our brains. Once our brains have developed sufficiently, which happens pretty early on, we don't make more neurons, and those have to last a lifetime. Hopefully they will. And, unless you're developing a dementia or having a serious accident like a stroke damaging your brain, hopefully those neurons will last for a lifetime. Your muscles also, eventually you stop getting new muscle cells, and instead, if you're working out and you're getting fitter, what you're doing is making the existing muscle cells bigger, but you're not actually developing new muscle cells. But other tissues actually do go on doing mitosis throughout your life. Can you think of tissues in the body where we keep making new cells?
Liver cells?
I'm sorry, where?
Liver?
Well, liver. I'm not sure there, actually. I'd have to look at that, probably. But I was thinking more like skin being an obvious one. We're all shedding 1 billion or so skin cells every day. That actually becomes a large part of house dust. So if you're vacuuming the house, a lot of what you're vacuuming up is actually yourself. Kind of disgusting to think about, but it's true. So we are shedding billions of skin cells every day, or at least every week, and we're constantly growing new ones. We're doing that also in, for instance, in your digestive tract. You're making billions of new cells lining the digestive tract. And we heard liver, and I think that is true. Also bone. If you are putting bone under stress, like you're doing a lot of exercise, you will grow new bone cells. The bones will get thicker. But certain other areas, these divisions stop happening. So the idea that you keep doing mitosis through your life, well, you do, but it gets to be just in certain areas. Also, this kind of division helps for asexual reproduction. In a lot of species, we get a lot of fungi, for instance, that reproduce asexually. They'll also often have a sexual phase, but not always. And plants can sometimes do that, making new plants from small fragments. And even some animals can do that. We don't think about animals reproducing without sex, but there are cases where that happens. So here's with plants, where you have here these stands, these forests of aspen trees in the Rocky Mountains. And when you've got a lot of them growing in an area like this, they're all clones, they're all copies. You can be standing under this whole canopy of aspen trees, and all the trees that are around you are exact copies genetically of each other. They're all connected underground. At one point, maybe thousands of years ago, an aspen would have gotten started from a seed, and it grew up, and that one tree didn't live very long, maybe 50 years. But then it put out roots, and the roots started putting up new growth through the surface. The roots will grow down, but with the aspens, they also grow up, and they produce these things called suckers that then develop into new trees. So the aspen can take over a whole hillside, given time, thousands of years, for one of these big clones of aspens to be in one area. These are all reproducing without sex. And sea anemones can do the same thing. Here we have these sea anemones that are showing up on rocks that are in the intertidal area, close to shore. And this one, you can see, is actually splitting off to produce 2 clone daughters, starting from one. That one sea anemone then got extra big, and then now it's dividing, looking almost like 2 cells dividing, but it's actually 2 animals forming from one, because these are actually very primitive animals. And what you will get are areas around Puget Sound where we have thousands of these that are on the rocks, and they're all in contact with each other. And sea anemones are territorial. If one of them touches a different sea anemone and it's not a clone sister, then they will attack each other. They will try to kill each other to take over that rock. Sometimes they will kill each other by stinging back and forth with these poison-tipped tentacles that they have. But if one of these touches a clone sister, that's all fine. They like each other, long as they're copies. So Mitosis is doing a lot of things for us, and some of that is asexual reproduction. Doesn't happen with us. But here's where you'll get the massive number of sea anemones all together, and they're coexisting peacefully because they recognize each other as being the same. Now, we get a division happening in bacteria. We call that prokaryotic fission. Bacteria, you will do something that's a little bit like mitosis, where one cell produces two daughter cells that are copies. The mechanics of it, though, are very different from mitosis, so we don't call this mitosis. Even though the end result is different, you get there by a totally different mechanism. So we won't be looking at this further. But that's what happens with the prokaryotes, things like bacteria. Looks something like this when you have an actual bacterium photographed as it's dividing. It has made it so that we have two exact copies of the bacterial DNA in both of these cells, and now that is pinching off to form two separate cells. Bacteria can sometimes do this in 20 minutes. They're extremely fast at reproducing. Some of them are. So, anyone have questions on this so far? Okay, let's look at it some more. So, when we're talking about eukaryotic cells, like our cells, we go through this thing called the cell cycle. This assumes that a cell is continuing to divide. That is one that will continue to be producing daughter cells, and they will keep dividing and making more daughter cells, like, for instance, in your skin. So what you're doing with some stem cells that are in your skin is that you go through a division, what we call M phase or mitotic phase. One cell splits to make two cells. And then if we just track one of those cells, eventually it goes through this long period we call interphase when it's not dividing, and eventually it gets around to where it can divide again. So cells alternate between dividing and not dividing. When they're dividing, we say they're in mitotic or M phase. When they're not dividing, they're in interphase. Now, what's happening is that when you produce a daughter cell, it has one copy of all the DNA molecules, all the chromosomes, and it goes into what we call the G1 phase, or first gap. And this is when it's just growing, because it's starting out too small. It's growing, doing things like making more mitochondria, making more ribosomes, making more of everything, getting bigger, and doing whatever its function is. G1, the cell is just doing whatever it's there for. And all that time, it just has one copy of its chromosomes. At some point, the cell will be signaled to get ready to divide again. If it's still in the cell cycle, it should eventually get a signal telling it to divide. And what it will do is enter into S phase. That is synthesis of new DNA. That's the fundamental thing the cell has to do before it can divide again. It has to copy all the chromosomes that have the DNA in them. So it will spend a while duplicating all of the chromosomes, all the DNA molecules. Following that, it'll go through a second gap period when it's finishing preparations to divide. So it's going through other preparations to divide, like making more proteins, making microtubules, which it involves in separating, those are involved in separating the chromosomes, doing a lot of stuff to get ready to divide. Then it goes into M phase, it divides, and then we're back to the first gap. So we're going from one copy of the chromosomes. As we go through S, we're going from 1 to 2 copies, and then all the way through G, we have 2 copies of the chromosomes, and we go into mitosis like that, and partway through, we go back down to 1 copy of each chromosome in each cell. And then we're back to 1 copy for a while, for an indefinite period. G1 could last a long time. Maybe the cell isn't supposed to divide again for a few months. So the G1 might last for a long time, until the cell is signaled to divide again. Then the S and the G2 phases happen in a fairly standard amount of time. They're fairly fast. Mitosis takes a fairly standard amount of time to happen. G1 might take a very long time, might stay in G1 for an extended time. So that's an overview of the cell cycle. Is this making any sense here so far?
Yeah.
Any of it not making sense?
How long does the mitosis take? How long does it take, roughly?
Well, we're actually going to see an example of that later. We're talking hours possibly for mitosis to happen, maybe up to a day. So I will show you some examples of it happening later. I think we're basically talking hours for mitosis to happen. Other questions? Okay, well, you have these chromosomes, and each chromosome, this is important to keep in mind, each chromosome has one DNA molecule and thousands of proteins that are helping to organize it. So it's a very, very large macromolecule. The chromosome has one DNA molecule, which probably has hundreds of millions of nucleotides in it. They're extremely large macromolecules. Then the single DNA molecule in a chromosome is combined with the thousands of proteins. What we're seeing at the right here, this is one of the chromosomes that's been duplicated. So this is going through the S phase of interphase. The DNA is getting duplicated. You end up with chromosomes that look like this under the microscope. You have two exact copies joined together. They're joined at this little place where they pinch together called the centromere. But basically, this is one DNA molecule that's tightly wound up and bundled with proteins. And this on the right is an exact copy of that same DNA molecule. When the cell splits, one of these goes into one of the daughter cells, the other copy goes into the other daughter cell. And something very important to memorize is that human cells have 46 chromosomes. That's the standard number for humans, 46. We'll see later how sometimes people have different numbers, but nearly everybody is going to have 46 chromosomes in their cells. This is ignoring the mitochondrial DNA, which is just a tiny amount of DNA. With all of this, with mitosis, we're talking about nuclear DNA. So you'll have a chromosome basically consisting of a DNA molecule along with proteins that we're not looking at here. And as we go through the cell cycle, those DNA molecules will get duplicated and you will have two exact copies of the chromosome joined together. We say, as we go from this chromosome to this, we say it's still one chromosome. What we have now are two sister chromatids that are joined together. They are two exact copies of the same chromosome. And we say this is still a chromosome rather than two. The way to think about that is you've got exactly the same amount of information, whether it's one copy or two copies. It's like if you have, say, a music file of one song and then you get a backup copy of it, you still just have the one song. It doesn't matter if you have more than one copy of it. And that's something like what happens here. So chromosomes with human cells, we stay at that 46 count as the cells are going through their cell cycle and dividing. 46 remains constant. Only question is whether the chromosomes are unduplicated or duplicated. So you start with the chromosomes being unduplicated, and then you duplicate them, and these separate copies go into different cells, and you're back to unduplicated once again. Is this making sense so far?
Yep, so far so good.
Thank you. So what we're seeing here at the right, this is a set of the human chromosomes. It's all been colorized, and they don't usually look like this, where they look like little sticks or worms. During most of the cell cycle, you're not going to see the chromosomes looking this way, but during mitosis, the chromosomes will condense into very compact structures, and then they actually look kind of like we have here, except for the kind of psychedelic coloring that was put on these. The chromosomes we're looking at here, these are numbered from biggest to smallest. This is number 1 at the upper left, and it goes through the numbers, finally getting to number 22 right here. And then we have a pair of sex chromosomes. These are 2 X chromosomes. So did this come from a boy or a girl? Any guess?
Boy.
Oh, no. Girl. Yes. Women have 2 identical X chromosomes, or actually not identical, but they're the same size as each other. 2 X chromosomes, and they're both pretty big. Men get an X and a Y. The Y chromosome is about as big as one of these little ones, so men don't get as much DNA. There are actually some health consequences of that. There's a few diseases that men tend to get, and it's because they have a little Y instead of a big X. So we'll look some at that later. But again, the chromosomes can be unduplicated, duplicated, and when they are duplicated, we say it's still one chromosome consisting temporarily of 2 sister chromatids. Now, the DNA that's in the chromosome can be super highly condensed. And right here, we're seeing some of that. We have DNA that's being shown here with the twisty double helix. It gets wrapped around balls of proteins called histones. And you'll have these histones and a little bit of DNA going in between. And the DNA can be pretty loosely organized like we have here toward the bottom, or it can get coiled up into bigger and bigger supercoils, eventually becoming the supercondensed chromosomes that we see during mitosis. Most of the time during the cell cycle, the DNA is not supercondensed and distinguishable like we see here at the upper left. Instead, it's more disorganized, more like what we have here toward the bottom. Well, this X shape of the chromosomes shows up at the beginning of mitosis. That's where we have 2 duplicated chromosomes, and the chromosomes have condensed due to interactions with a lot more proteins until the DNA becomes super condensed, like we see in the case of this X pattern. During interphase, the DNA is not as compacted as that, and you can't see the chromosomes distinctly. So during mitosis, we are able to see the chromosomes as distinct, separate shapes because their DNA is condensed and become very tightly packed. Except when you're not in mitosis, then you can't see the DNA, and the nucleus looks more like a gray blob. It doesn't have distinct structures in it. So we often will see these diagrams where chromosomes look like we have here, or maybe more like over here at the left. But you only see the chromosomes like that when the cell is actually busy dividing. Are you with me on that?
Yes.
Okay, so we have interphase. And with interphase, we have those 3 parts we already looked at on that big cycle diagram. A cell is newly formed, a new daughter cell is formed, and initially it just has 1 copy of all the chromosomes. It enters into a period of just normal growth, normal function, which may go on for a long time. That's G1. During G1, it just has 1 copy of every gene, every chromosome. But again, if it receives a signal telling it to divide, the first thing it has to do is copy all the genes, because it's going to make 2 new cells, and each new cell needs a copy. So it gets busy copying the DNA. Eventually ends up where instead of having 46 DNA molecules, how many does it have? At the end of S phase, how many DNA molecules does it have if it's a human cell? Is it 46?
Yes, isn't it always 46?
Well, that's how many chromosomes, but how many DNA molecules does it have?
Right.
Yes. Going to be a little more than 46. Twice as many, actually, 92.
92.
That's how many DNA molecules. But we say it's still 46 chromosomes because it's really just pairs of identical copies joined together. And making copies doesn't mean the cell has any extra genes. It doesn't know how to do any new things. So we start with 46 chromosomes, single copies. We go through S phase. At the end of that, we've got 46 duplicated chromosomes. G2, all the way through there, we still have 46 duplicated chromosomes. And the cell is generally getting bigger, making proteins that'll be involved in mitosis, getting ready to divide. And during this entire period, the chromosomes don't stand out under the microscope. The nucleus just looks like kind of a blob without any features in it. Now, here's something too with the terminology. Some cells stop dividing. That would be like the mature brain cells. We say that those have left the cell cycle and they've gone into what's called the G0 phase. They've exited the cell cycle. They've left it behind. They are not going to divide anymore. That happens with some of our cells. But we are concentrating on cells that are staying in the cell cycle. Mitosis is still in their future. So, interphase, this is an onion cell. So this is in a plant root. And this is an area where there's a lot of division happening when you're in a root. You have a growing tip of the root. It's growing rapidly into the soil. And these cells are dividing rapidly. We will see examples of these later. And this cell is in interphase, and its chromosomes are not really standing out. We are seeing one detail in the nucleus. Does anyone remember where the nucleus has an organelle inside of it called the nucleolus? That's inside the nucleus. It's one of the things that we looked at briefly in your chapter 4. And this has an important function. If you're in an interphase cell, the cell has just formed from a parent cell that's split, and that split up the cytoplasm. That means it also split up the ribosomes into the two daughter cells. And a cell might need 5 million ribosomes to function. Well, after splitting, say it has 2.5 million, it gets busy making new ribosomes, and that's what this nucleolus is for. That's the ribosome factory. You have genes on some of your chromosomes that tell you how to make copies of all the parts of the ribosomes. This is an area where that activity is going on very intensively. So the cell can make millions of new ribosomes since it doesn't have as many as it used to. Beyond that, we're not really seeing details in the nucleus, not the way we will later. But that's an interphase cell. The cell is just growing, doing whatever it's there for. It might be duplicating the DNA. We wouldn't be able to tell from looking at it under the microscope. The chromosomes are not distinct. And we say, in this case, that this is chromatin. This is just loosely packed DNA. And you can't look at it at one spot and say, ah, there's the double helix. Are you with me on this?
Yes.
Well, we're just going to do this for a few more minutes, and actually, let's maybe say that's enough right there. Let's do that. Let's say that this is far enough because we're already ahead of schedule. So I'm going to say it's okay for you guys to take off.
Awesome. Thank you so much.